Eat these 3 foods before bed to make new mitochondria. The quest for a longer, healthier life has captivated humanity for millennia, but only in recent decades have we begun to unravel the intricate biological mechanisms that govern aging. At the forefront of this groundbreaking research stands Dr. David Sinclair, a distinguished geneticist and professor at Harvard Medical School, whose work has profoundly influenced our understanding of how we age and, more importantly, how we might intervene to extend our health span, the period of life spent in good health. Dr. Sinclair's research often centers on the idea that aging is a disease, or at least a treatable process, rather than an inevitable decline, and that we possess significant power to influence this process through lifestyle choices, including diet. Central to his theories and much of modern longevity science is the role of mitochondria, often referred to as the powerhouses of the cell. These tiny bean-shaped organelles are indispensable for virtually every biological process, converting the food we eat into adenosine triphosphate, ATP, the primary energy currency of the cell. Without robust, efficient mitochondria, our cells cannot function optimally, leading to a cascade of issues that underpin many age-related diseases, from neurodegeneration to metabolic dysfunction. Understanding the profound importance of mitochondria is the first step towards appreciating the impact of specific dietary choices on our cellular health. Mitochondria are more than just energy factories. They are intricate regulators of cellular metabolism, signaling, and even programmed cell death. They produce the vast majority of our cellular energy through a process called oxidative phosphorylation, a marvel of biological engineering. However, this process is not without its costs. It also generates reactive oxygen species, ROS, often called free radicals, as byproducts. While a certain level of ROS acts as important signaling molecules, an overload can lead to oxidative stress, damaging cellular components, including the mitochondria themselves. This vicious cycle of damage and dysfunction contributes significantly to the aging process. Healthy aging, therefore, hinges critically on maintaining mitochondrial function, protecting them from damage, and crucially, stimulating the creation of new, healthy mitochondria, a process known as mitochondrial biogenesis. When our bodies can efficiently produce new mitochondria and clear away old, damaged ones, a process called mitophagy, our cells maintain their vitality, resilience, and capacity to perform their myriad functions. Dr. Sinclair and other longevity researchers emphasize that boosting mitochondrial biogenesis and function is one of the most powerful strategies we have for extending health span and delaying the onset of age-related decline. This leads us to the exciting prospect that certain foods, particularly when consumed strategically, might offer a direct pathway to enhance this fundamental cellular process. The idea that specific nutrients consumed before sleep can effectively signal our bodies to build more and better mitochondria is a fascinating and empowering concept, suggesting that our nightly routine could become a potent tool for cellular rejuvenation. So, what are these specific foods that Dr. David Sinclair and the broader scientific community suggest can stimulate mitochondrial biogenesis, especially when incorporated into your pre-sleep routine? While Dr. Sinclair himself might recommend specific compounds like resveratrol or NMN, the underlying principle is that certain whole foods contain powerful phytonutrients that can activate the same cellular pathways targeted by these supplements. These pathways include sirtuins, a family of proteins that play a critical role in cellular health and longevity, AMPK, AMP-activated protein kinase, a master regulator of energy metabolism, and NAD+, nicotinamide adenine dinucleotide, a coenzyme vital for cellular energy and repair. By activating these pathways, we can trigger the upregulation of PGC1-alpha, a master regulator of mitochondrial biogenesis. Let's delve into 3 categories of foods that consistently appear in discussions around mitochondrial health and longevity, and why consuming them before bed might amplify their benefits, transforming your evening snack into a powerful cellular rejuvenation strategy. the first category of food that holds immense promise for mitochondrial health and which aligns with the principles often discussed in longevity circles, is dark berries, such as blueberries, raspberries, and blackberries. These vibrant fruits are not merely delicious. They are a concentrated source of some of nature's most potent compounds. Their deep hues are indicative of their richness in anthocyanins, a class of flavonoids known for their powerful antioxidant and anti-inflammatory properties. Beyond anthocyanins, berries are also abundant in other beneficial polyphenols, including quercetin and ellagic acid. When we consume these berries, we're introducing a complex symphony of bioactive molecules into our system. These compounds don't just scavenge free radicals, they actively communicate with our cells, signaling them to enhance their protective and regenerative capabilities. Research suggests that the polyphenols in berries can directly activate sirtuins, particularly SIRT1, which is a key player in promoting mitochondrial biogenesis and DNA repair. By mimicking the effects of caloric restriction, these compounds encourage cells to prioritize maintenance and repair over growth, a fundamental strategy for longevity. Furthermore, berries have been shown to boost AMPK activity, which then signals the cell to produce more mitochondria and improve their efficiency. The before-bed timing for berries is particularly interesting. Their relatively slow digestion, combined with the body's natural restorative processes during sleep, could allow for a sustained release and absorption of these powerful phytonutrients, providing a consistent signal for cellular repair and mitochondrial generation throughout the night. Moreover, some berries like tart cherries contain natural melatonin, which can support restful sleep, and quality sleep itself is a critical component of cellular repair and mitochondrial health. Consuming a handful of fresh or frozen mixed berries an hour or two before bed, perhaps sprinkled over a small serving of plain Greek yogurt or mixed into a smoothie, offers a delicious and scientifically grounded approach to supporting your cellular powerhouses. The antioxidants in berries can also help mitigate any oxidative stress that might accumulate during the day, preparing your cells for optimal repair and renewal cycles while you rest. The cumulative effect of regularly integrating these nutrient-dense fruits into your evening routine could be substantial, contributing to a more resilient and energetically robust cellular infrastructure over time, directly supporting the ambitious goals of health span extension championed by Dr. Sinclair. The second crucial category of foods for mitochondrial health, especially impactful when considering a before-bed consumption strategy, consists of entire cruciferous vegetables like broccoli, kale, spinach, and Brussels sprouts. These vegetables are absolute powerhouses of unique sulfur-containing compounds called glucosinolates, which, when the plant cells are damaged, e.g. by chewing or chopping, are converted into biologically active compounds like sulforaphane and indole-3-carbinol, I3C. These compounds are not merely antioxidants, they are potent activators of the NRF2 pathway, a master regulator of cellular defense against oxidative stress and inflammation. The NRF2 pathway is incredibly important for mitochondrial health because it upregulates the production of endogenous antioxidant enzymes, such as glutathione and superoxide dismutase, which are far more effective at neutralizing free radicals than dietary antioxidants alone. By enhancing our internal antioxidant defense systems, sulforaphane and its relatives directly protect mitochondria from damage, thereby preserving their function and integrity. Furthermore, cruciferous vegetables contain other beneficial compounds like quercetin and K-empherol, which also contribute to their anti-inflammatory and mitochondrial supportive effects. Some research also suggests that compounds in cruciferous vegetables can influence sirtuin activity and support detoxification processes that indirectly benefit mitochondrial function by reducing cellular burden. Consuming these vegetables before bed, either lightly steamed, roasted, or even as part of a small salad, can provide a steady supply of these NRF2 activators as your body enters its repair phase. The digestive process slowly releases these compounds, potentially leading to prolonged activation of protective pathways. The fiber content in these vegetables also supports gut health, which is increasingly understood to be intertwined with overall metabolic and cellular health, including mitochondrial function. A healthy gut microbiome can influence the absorption of nutrients and the production of beneficial metabolites, all contributing to a cellular environment conducive to robust mitochondrial biogenesis and repair. While some might worry about gas or indigestion from raw cruciferous vegetables before bed, lightly cooking them can make them easier to digest while preserving many of their beneficial compounds. A small portion of steamed broccoli or sautéed spinach as part of your evening meal, or even as a standalone snack a couple of hours before sleep, can provide a steady flow of these protective compounds, giving your mitochondria the support they need to work efficiently and to regenerate throughout the night. This strategic consumption leverages the body's nighttime repair cycles, transforming a simple vegetable into a potent tool for cellular longevity, aligning perfectly with Dr. Sinclair's emphasis on proactive biological maintenance, the third and equally vital category of foods that can significantly contribute to mitochondrial health, with compelling reasons for pre-sleep consumption, encompasses healthy fats, particularly those found in avocados and extra virgin olive oil. These foods are rich in monounsaturated fatty acids, MUFAs, primarily oleic acid, which are crucial for maintaining the fluidity and integrity of mitochondrial membranes. The mitochondrial inner membrane, where the electron transport chain resides, is highly specialized and relies on a specific lipid composition to function efficiently. Healthy fats provide the building blocks for these membranes, ensuring they remain pliable and capable of facilitating the complex biochemical reactions required for ATP production. Beyond their structural role, these fats also carry powerful fat-soluble antioxidants like vitamin E, especially gamma-tocopherol in olive oil, and a range of polyphenols. Extra virgin olive oil in particular is celebrated for its unique polyphenols, such as oleocanthal and hydroxytyrosol. Oleocanthal has anti-inflammatory properties similar to ibuprofen, while hydroxytyrosol is an extremely potent antioxidant. These compounds actively combat oxidative stress that can damage mitochondria, preventing the vicious cycle of ROS-induced dysfunction. Furthermore, some research indicates that polyphenols found in olive oil can influence sirtuin activity and modulate pathways related to inflammation and cellular stress response, indirectly promoting a healthier environment for mitochondrial function and biogenesis. The rationale for consuming these healthy fats before bed is multifaceted. Firstly, healthy fats promote satiety, which can help prevent late-night cravings for less healthy options, thus supporting overall metabolic health. Secondly, their slow digestion and absorption can provide a sustained release of their beneficial compounds throughout the night. The anti-inflammatory and antioxidant effects can continue to protect cellular structures, including mitochondria, during the long hours of sleep when the body is engaged in extensive repair and regeneration. A small portion of avocado, perhaps mashed on a whole grain cracker or simply eaten sliced, or a tablespoon of high-quality extra virgin olive oil drizzled over a small portion of vegetables or a protein source, can serve as an excellent pre-bed snack. The presence of healthy fats also supports the absorption of fat-soluble vitamins, like vitamin E, and other beneficial compounds present in other longevity foods. By providing the essential structural components for mitochondrial membranes and delivering a continuous supply of anti-inflammatory and antioxidant compounds, avocados and extra virgin olive oil act as vital guardians and builders of your cellular powerhouses. This strategic incorporation of healthy fats into your evening routine is not just about avoiding unhealthy snacks. It's about actively fueling your cells with the precise nutrients they need to optimize mitochondrial performance and regeneration, a cornerstone of Dr. Sinclair's vision for combating aging at its cellular roots. The scientific proof supporting the claims about these foods and their impact on mitochondrial health is robust and growing, forming a foundational pillar of modern longevity research. The mechanisms through which these foods exert their beneficial effects are not speculative. they are increasingly being elucidated at a molecular level, echoing the very pathways Dr. David Sinclair has dedicated his career to studying. The activation of sirtuins, particularly SIRT1 and SIRT3, is a recurring theme. Sirtuins are a family of NAD+ dependent deacetylases that respond to cellular energy status. When energy is low, mimicking caloric restriction, NAD+ levels rise, activating sirtuins. This activation triggers a cascade of events that promote cellular repair, stress resistance, and, crucially, mitochondrial biogenesis. Many polyphenols found in berries, cruciferous vegetables, and olive oil are known sirtuin activators, or indirectly support their function by improving cellular NAD+ availability or reducing oxidative stress that can impair sirtuin activity. For instance, resveratrol, a well-known sirtuin activator, is found in grapes and thus wine, and its molecular cousins and synergistic compounds are present in high concentrations in berries. Another critical pathway is the AMP-activated protein kinase, AMPK. AMPK acts as a cellular energy sensor. When ATP levels drop and AMP levels rise, AMPK is activated. This activation signals the cell to shift from energy-consuming processes to energy-producing ones, which includes increasing mitochondrial biogenesis and fat oxidation. Many compounds in the aforementioned foods, such as quercetin in berries and certain compounds in cruciferous vegetables, have been shown to activate AMPK. By stimulating AMPK, these foods encourage the cell to produce more mitochondria and improve their efficiency, akin to the metabolic adaptations seen with exercise and caloric restriction. The NRF2 pathway is another central player. NRF2 is a transcription factor that regulates the expression of a vast array of antioxidant and detoxifying genes. Sulforaphane from cruciferous vegetables is one of the most potent known activators of NRF2. By boosting NRF2 activity, these foods enhance the cell's endogenous defense mechanisms, significantly reducing oxidative stress on mitochondria. This protection is vital because damaged mitochondria are less efficient and can themselves become sources of further oxidative stress, creating a vicious cycle of decline. By bolstering these defenses, we help maintain the integrity and function of our existing mitochondria while also creating a healthier environment for new ones to form. Furthermore, these foods contribute to mitochondrial health by influencing mitochondrial dynamics, which refers to the continuous processes of fusion and fission. Fusion allows mitochondria to merge, sharing resources and repairing damaged components, while fission creates new, smaller mitochondria or segregates damaged parts for removal. Healthy fats contribute to the fluidity of mitochondrial membranes, which is essential for these dynamic processes. Many phytonutrients also promote mitophagy, the selective degradation and recycling of damaged or dysfunctional mitochondria. This quality control mechanism is crucial for removing inefficient powerhouses and making way for new, healthy ones, preventing the accumulation of cellular debris that contributes to aging. The before-bed timing amplifies these benefits by capitalizing on the body's natural circadian rhythms and sleep-related restorative processes. During sleep, the body undergoes significant cellular repair, waste removal, and hormonal regulation. Consuming nutrients that support mitochondrial health before this period of intense repair provides the body with the necessary building blocks and signaling molecules precisely when they are most needed. The slow digestion of whole foods, especially those rich in fiber and healthy fats, ensures a sustained release of these beneficial compounds throughout the night, providing a continuous signal for mitochondrial biogenesis and protection. This strategy is not about immediate energy, but about long-term cellular programming and support. It transforms a simple evening habit into a potent, proactive health strategy, aligning perfectly with Dr. Sinclair's message that we can actively intervene in the aging process through informed lifestyle choices, giving our cells the best possible chance to thrive and renew themselves as we rest. Implementing these dietary strategies into your routine doesn't require drastic changes. Rather, it encourages mindful and consistent choices. For dark berries, aim for about a half to 1 cup of fresh or frozen berries. They are incredibly versatile. You could add them to a small bowl of plain Greek yogurt for added protein, blend them into a pre-bed smoothie with a handful of spinach and a splash of unsweetened almond milk, or simply enjoy them on their own. Freezing berries actually helps to break down their cell walls, potentially making some of their beneficial compounds more bioavailable. For cruciferous vegetables, a smaller portion, perhaps half a cup, is sufficient. Lightly steaming broccoli florets or sautéing a handful of spinach or kale with a touch of extra virgin olive oil are excellent options. The key is to avoid heavy, rich preparations that might hinder sleep. Simplicity and digestibility are paramount for a pre-bed snack. As for healthy fats, a quarter of an avocado sliced or a tablespoon of high-quality extra virgin olive oil drizzled over your chosen vegetable, or a small piece of lean protein like baked fish can provide the necessary beneficial lipids and polyphenols without being too heavy. The overarching goal is to integrate these foods as part of a balanced, whole foods-based diet. While these specific foods are highlighted for their direct impact on mitochondrial health, their benefits are amplified when consumed within a broader dietary pattern that emphasizes unprocessed ingredients, abundant plant matter, lean proteins, and minimal added sugars and unhealthy fats. Hydration is also crucial. Drinking enough water throughout the day supports all cellular functions, including those of the mitochondria. Furthermore, remember that diet is only one component of a holistic longevity strategy. Dr. Sinclair and other experts consistently emphasize the synergistic effects of various lifestyle interventions. Regular physical activity, particularly a mix of endurance training and high-intensity interval training, HIIT, is a powerful stimulator of mitochondrial biogenesis and function. Exercise creates a cellular demand for energy, prompting the body to produce more mitochondria to meet that demand. Intermittent fasting or time-restricted eating can also be profoundly beneficial, as periods of fasting activate many of the same longevity pathways as caloric restriction, including sirtuins and AMPK, and can enhance mitophagy, clearing out old, damaged mitochondria. Quality sleep, beyond just the timing of nutrient intake, is itself a non-negotiable pillar of cellular repair and rejuvenation. During deep sleep stages, the brain clears metabolic waste and the body undertakes extensive cellular maintenance, processes that are heavily reliant on well-functioning mitochondria. Stress management techniques, whether through meditation, mindfulness, or spending time in nature, also contribute significantly by reducing chronic inflammation and cortisol levels, both of which can negatively impact cellular health and mitochondrial function. In conclusion, the message from Dr. David Sinclair and the burgeoning field of longevity science is one of empowerment and agency. We are not passive spectators in our aging process. We have powerful tools at our disposal to influence our health span at a cellular level. By strategically incorporating these three categories of foods, dark berries, cruciferous vegetables, and healthy fats like avocado and extra virgin olive oil into our pre-sleep routine, we are actively supporting the creation of new, healthy mitochondria. We are signaling our bodies to activate critical longevity pathways like sirtuins, AMPK, and NRF2, while also providing essential building blocks and protective compounds. This isn't about a magic pill. It's about leveraging scientific understanding to make informed, consistent choices that collectively contribute to a more resilient, energetic, and youthful cellular environment. By making these small, delicious changes to our evening routine, we are not just eating before bed, we are investing in our cellular future, one nourishing bite at a time, transforming our sleep into a powerful period of profound cellular renewal and rejuvenation, thereby taking a proactive step towards a longer, healthier life.